morning to everyone. I hope everyone can hear well. My name is Pema Yangzom and I'm from Bhutan. I'm doing my MSc in dentistry at Konkan University. And I would like to present our study on characterization of Asian oral cancer spheroids using the liquid overlay technique. This is my outline. Head and neck cancer is the sixth most common cancer in the world. According to an epidemiology report, there has been increasing incidence rate of oral and lip cancer in the South and Southeast Asia, and the 5-year survival rate is only 50%. Despite the different treatment modalities, there is no improvement in its prognosis because of several factors, including chemotherapy resistance, and the resistance is due to the complex tumor microenvironment and tumor cell heterogeneity, which is not represented well by 2D cell culture, as it has reduced cell-to-cell and cell-to-ECM interaction. So, to recapitulate, the complex in vivo microenvironment, 3D cell culture is necessary. As it has dynamic cell-to-cell and cell-to-ECM interaction, it represents partial arrangement of different cells with different morphology, differentiation, proliferation, gene expression, and similar growth kinetics to those of tumor in vivo. If we have a look at the microenvironment of tumor in vivo, we can see that the cells proliferate, and with time, they become compact, and they develop nutrition gradient like this, whereby it's minimum at the center and high at the periphery. Hence, they develop necrotic cells at the center, surrounded by the quiescent cells, and then actively proliferating cells at the periphery. Similar microenvironment is observed in 3D tumor in vitro, where we have single cell suspensions immersed in growth medium, and with time they grow and they become compact like this, and they too develop the nutrition gradient, and they have necrotic cells at the center, surrounded by the quiescent cells and the proliferating cells at the periphery. Now, to have a look at the techniques to carry out 3D cell culture, we need to understand 4 types of spheroids. Let me remind, a spheroid is a well-rounded 3-dimensional tissues. The first one, multicellular tumor spheroids, or MCTS, has 3 techniques, and the suspension culture, the first one, is further divided into 3, which I'll explain later in my next slide. We have gel embedding culture, scaffold culture, and the rest of 3. And if you focus on the suspension culture, the first we have is a liquid overlay technique. In this one, we try to grow the spheroids or 3D tissues in a non-adherent condition, whereby we coat the surface of the culture plate with a non-adherent polymer and allow this 3D tissues to form. Likewise, we have hanging drop technique and the device-assisted technique, which are of these 4 types. Now, if you compare all these techniques, you can see that to grow MCTS, the techniques for MCTS has good success rate of initiation, tumor-stroma interaction, and characteristics of original tumor, and very good ease of maintenance, tumor growth, and high throughput drug screening. And if you compare within the MCTS techniques, the liquid overlay technique is cheapest, very simple method, and easy access to spheroids, and very compatible with high throughput drug screening. And if you look at the literature, we can see that this paper used liquid overlay technique to successfully grow the MCTS of head and neck squamous cell carcinoma cell lines. In particular, if you see ORL48, it is a human oral squamous cell carcinoma cell line. It has been grown with gel embedding technique and flicking technique. And there's another cell line called ORL136, and ORL136 and ORL48 are both oral squamous cell carcinoma cell lines. And in literature, they never grew spheroids of these 2 cell lines using liquid overlay technique. Hence, the objectives of my study are to successfully create spheroids of ORL48 and ORL136 using liquid overlay technique to determine its characteristics, and then to compare the difference in the morphology within the same spheroid and the different cell lines on different days of cell culture. So it is an experimental research whereby I grow these 2 cell lines as 2D first, and then I'll use liquid overlay technique to grow as 3D. And then I compare their morphology based on shape, size, surface, and growth. For 2D cell culture, these 2 cell lines are obtained from Head and Neck Cancer Research Team, Malaysia. Firstly, we grow them as single cell suspension, and then we incubate it, and we wait until they become 80 to 100% confluency. After reaching the 80 to 100% confluency, we trypsinize it, and we calculate the cell concentration to be 10,000 cells per well and 25,000 cells per well in 200 microliters of DMF12 complete medium. For that, we use U-bottomed 96-well plate, and then we pre-coat it with 1% agarose, and we allow to dry it for 30 minutes to 1 hour under the hood. And then after cell seeding, we have to centrifuge it for 10 minutes at 1000 RPM, and then we incubate it for 7 days for them to form 3D tissues. And to compare the morphology, we use phase contrast microscopy and we observe until day 7. And then to carry out this replating of MCTS, what we do is we transfer the MCTS of both the cell lines from U-bottom 96-well plates to flat bottom 96-well plates on the 7th day by using wide bore pipette tip, and then we observe it under the phase contrast microscope. Now let me tell you, this replating we carried out because to see that whether the spheroid remain intact or not after we transfer to the flat bottom 96-well plates. For H&E staining, first we wash the MCTS of both the cell lines with PBS, and then we fix it with 4% paraformaldehyde overnight, and we transfer it to Eppendorf tubes, and then we remove the paraformaldehyde, and we followed by PBS wash, and then we add 0.35% agarose, and along with the agarose solution, we transfer them to 6-well plates, and the spheroid looks like this. They look suspended in agarose gel, and then we send them for H&E staining. For data analysis, we use ImagePro Plus 7 and descriptive statistics for analyzing the data. Now the results. This table, you can see that we have 4 different conditions. The first 2 rows represents 10,000 cells per well for ORL48 and ORL136. The third and fourth row represents 25,000 cells per well for both the cell lines. And if you look overview, right, we can see that we formed a single well-rounded MCTS with smooth periphery for all the 4 conditions. And then if you compare the first row with the second row, you can see the diameter is larger in the ORL48. Similarly, in the third and the fourth row, if you see the diameter is larger in ORL48 than ORL136. And if you look generally from day 1 to day 7, you can see that the diameter does not increase. It is either diminishing or remaining constant. Not only we had very good rounded MCTS, but we also had very few wells with irregular shape like this. And the third and the fourth row shows multiple spheroids, which later fuse together to become a single spheroid. And this is for 25,000 cells. If you look for 10,000 cells, we observe similar feature with irregular spheroid in the second row and multiple spheroid in the third row. And to compare the sizes, you observe from day 1 to day 7. And for 10,000 cells ORL48, the largest diameter is on day 1 and it keeps on decreasing until day 7. And similar trend is observed for 25,000 cells ORL48. And for 10,000 cells ORL136, we also see decreasing diameters with time. And if you compare it with ORL48, it has slightly smaller diameter. And for 25,000 cells ORL136, the diameter is decreasing. And if you compare it with ORL48, 25,000 cells, it is slightly smaller diameter. Overall, the smallest diameter recorded was 159.84 and the largest was 334.94 micrometers. Here we can appreciate the growth of the spheroids, the growth pattern, and you can see this diminishing growth with time, and ORL48 showed more diminishing growth than ORL136. And if you look particularly, the ORL136 is last line, for 10,000 cells, they more or less show like a dormant growth. And to see the core structure of the MCTS, these are H&E stained spheroids. You can see that there are 2 distinct regions. The outermost layer, which consists of 2 to 3 layers of squamous epithelial cells, and the inner densely packed squamous epithelial cells, which are dysplastic in nature. And then you can see 3 types or 3 states of cells as described in literature. The outermost, the 2 to 3 layers are called proliferating cells, and the inner one is called quiescent cells. And we can also observe few cells apoptotic, as well as some areas of disintegrated cells, which could be necrotic core. And this is the picture after replating on day 7, and you can see that the spheroid remained intact. It did not disaggregate into monolayer, hence a true spheroid. Now the discussion. Like the MCTS grown in literature, they have formed well-rounded spheroid, as you can observe in these 2 studies, and they also formed irregular spheroid like this. In particular, if you see ORL48, the spheroids, in the first paper, they have also irregular spheroid, and then in the second, they also have well-rounded spheroid. And I must say that it's comparable to our present study where we had well-rounded spheroid as well as irregular and multiple spheroids. And if you look at the size in literature, the smallest size is 46.76 micrometer and to maximum 400 micrometer. And it's comparable to our study, but it would depend upon the cell density, of course. The smallest is 159.84 and the largest being 334.94 micrometers. And if you look at the growth pattern, there are different growth pattern. It ranges from progressive growth towards rapid growth and moderate growth, and we have diminishing growth and static growth. So it would depend upon the cell line. And in particular, if you see MCTS of OL4A, this paper shows static growth and this paper shows progressive growth. But of course, they're using a different technique than ours. And our results also show that we had two types of growth, the diminishing and the dominant growth with time. And if you see the core structure, these are the core structure observed in Hiller spheroids, and they have a very remarkable necrotic core, as you can see, and very sparsely packed quiescent cells and thick proliferating layer. Compared to ours, of course, we have the proliferating layer at the periphery, and we have densely packed quiescent cells, and we have the unremarkable necrotic core. It could be because we are using different cell lines. And this did the replating of their spheroids on OL48, and they observed it from day 1 until day 4, and they observed that they disintegrated into monolayer from day 2 onwards, but they did the study to see the proliferative ability of the spheroids. While in our study, we just wanted to see whether the spheroids remain intact after we transfer to the flat bottom, and it remained intact. And this is my future work. I'll be testing the viability of the spheroids. And the first row represents OL48, where the green is for life, and it has no dead cells, and the host, and the composite of these three colors. And even OL136 shows similar trend. And the last row is for control, where the spheroids were treated with 70% ethyl alcohol. So the conclusion is that the MCTs of OIL48 and OIL136 were successfully generated using the liquid overlay technique, and the characteristics of the MCTs of both the cell lines were described based on shape, size, surface, and growth. And future work on cell viability, proliferation, and differential expression of molecular markers involving carcinogenesis of oral cancer, such as EGFR and E-cadherin, will be carried out. I'd like to thank Faculty of Dentistry, Konkan University, and TICA Foreign Ministry Thailand for their support. Thank you for your time and listening. Thank you.
Thank you very much for your presentation and very beautiful slides.
Thank you, Pa.
Well, we're gonna wait for the question from the audiences. Okay, maybe start from me. Well, it's very interesting. So you are the first liquid OLE technique to increase the number of the oral cancer, right?
Yes, Kajang, as per my knowledge.
So the question is, you just have the 7 day, right?
Yes.
7 day. So I want to know, do you experience any contaminations?
Oh, actually, I experienced contamination several times. Of course, it's a part and parcel of a laboratory cell culture. But I experienced particularly when I was growing them as monolayer, Ajahn. And but I was making sure that the 2D, the monolayer, the 2D cell culture was not contaminated before I do the cell seeding for 3D culture. So I didn't observe contamination in spheroids, but I did observe contamination in 2D cell culture.
So you're spheroid safe?
Yes.
Okay. Well, why you use the duration for 7 days?
Yes. Actually, there are various duration used in the literature. It could range from 3 days or 2 days to 7 days and to 15 days. But I use this 7 day in my study because the growth pattern of my spheroid, it's not progressively increasing. So that if it's progressively increasing, I would like to observe it for more and more days. But since it's like a diminishing pattern or it just remain constant, and then I try to check the viability as I've shown in my future work because it's diminishing in size. I was thinking maybe like it developed the necrotic core and it decreasing in size, but it seems that even observed the viability on day 7 and it doesn't have dead cells. So it's still viable at 7 day. And then thirdly, it matches to our objective because ultimately the goal of me growing as 3D is to serve as a suitable platform for testing the anti-cancer drugs. And we can successfully form the spheroid within 2 days, Kaja, which is suitable for testing the drug. So I thought 7 days is enough. I think keeping all those objectives in mind.
Well, I can see that you just count the amount of the spheroid, like 10,000, right, per well,
Yes,
right?
yes.
That your start cells, or you start from single cell and expand.
Oh, actually, in my pilot study, I started from 2,500 cells, and then 5,000. I varied the concentration from 2,500, 5,000, 10,000, 25, and 50,000. But initially, I used only 2,500, and it formed very small spheroid, and I just wanted to increase the size. So I chose 10,000, 25,000 cells for this preliminary study. But in my future work, I have involved all the variation of cells, starting from 2,500, minimum of 2,500 to maximum of 50,000 cells. And one more information, these huge numbers are like 10,000 and 25,000 are showing diminishing growth. But when I observed in 2,500 and 5,000 cells, it show increasing growth. So yes, this is the difference I observed.
So can I say like you found out the subcultures level, like it cannot exceeding 10,000. You have to do subculture to expand. Is that right? Or because it stopped expand, it's mean maybe
Yes.
subcultures.
Yes, yes, exactly. So I wanted to also, as Ajahn you clearly mentioned, like I wanted to get a threshold whereby it cannot increase. So I think 50,000 could be the threshold because it does not increase anymore. It keeps on decreasing. But with lesser number, it keeps on increasing. So for our study, in my opinion, I think, and as per the results, the threshold seemed to be 50,000 cells. But I didn't do more than that. I just limited to 50,000 cells.
50,000.
Yes, 50,000 cells per well.
But I can see like there are different between two strain.
Yes,
One strain is good at 20,000. The other strain is good at 10,000, right?
25,000.
25,000 and 10. So it's mean maybe the threshold of the subcultures might be different between two strains. Is that
Yes,
right?
it could be Ajan. And one important reason for that differences is that they are derived from people with different ethnicity, and they are derived from Ajan. OL48 is derived from a female gender, and it's from the gum, gingiva, and it is a stage 4 cancer. And the OL136 is derived from a male gender, and then it is from tongue cancer, and it's stage 1 Ajan. So this could reflect the differences.
Oh, that's okay. Thank you so much for your presentation,
Thank you.
Siti.